Okay, welcome to our Checking and Validating Your Wildlife Records webinar. So this webinar will be all about using the tool Record Cleaner to make sure that your data is in the correct format and to check for potential errors in the data as well. Um, so for those that don't know much about the Biological Recording Company, you can sign up to our free newsletter where we provide exclusive discounts for our webinars, etc. So well worth signing up to, and the link will be below the video in the chat. Coming up soon, if you're wanting more of an introduction to biological recording, I'd recommend our iRecord 101 webinar rather than this one. This one will be going into more detail about checking data. But if you want more information on the science of biological recording and how to use the iRecord tool, I'd recommend that. Again, the link will be in the chat or below the video if you're not watching this live. So, this is not biological recording 101. That's an error there. It's checking and validating your records. Session summary will be covering four different sections: a background to biological recording data, why it's important, and why it's important to make sure it's accurate. Then we'll be going into the validation of biological records before moving on to the record cleaner tool and how to use that yourself to validate your data. And then we'll be exploring how record cleaner can be used to support verification through the record cleaner rule system. Uh, More on that later, but those are the four sections that we're going to go through today. So, starting with the background to biological recording data, before I go on to this, maybe should say a little bit about who I am. So, my name's Kieran, and I run the National Recording Scheme for Earthworms. I'm passionate about biological recording and making sure that wildlife observation data is made accessible to the conservation sector for many different uses. So, wildlife observations and biological records, what are these? Well, they're synonyms for one another. The science of biological recording is the study of the distribution of living organisms. So, biological records describe the presence, abundance, associations, and changes, both in time and space, of wildlife. So, in their very purest form, the most basic form, they are the who, what, where, when of a wildlife observation. So the who being the person that is making that observation, the what being the organism that is being observed, the where being the location of where the observation took place, and when being the temporal information. So a very simple record might be me observing a fox outside of my flat in Denham with a grid reference so that we can locate that as a square, and then a date. And biological records, these wildlife observations, they go through a system we call data flow, and there are different components to that. So it starts off with the observation, actually seeing the or taking a photograph or hearing the organism. And then we go through creation where that is written down or entered digitally into a spreadsheet, etc. Then we go submission where it's that data is sent into a biological recording system. So in this current day and age, usually that is submitting it electronically somewhere, but that might have been posting off details of a record in the olden days. Then goes through a series of checks. So we've got validation where we check that everything's in the right format. We go through verification where we check that the data is likely to be correct. And then once it's been through those checks and any records that are not acceptable filtered out, we collate them into data sets where they can then be disseminated through platforms like the NBN Atlas, etc. There are lots of different organizations involved in biological recording, so biological recording networks. We can roughly break these down into four different groups. We've got local environmental record centres, which cover specific or defined geographic boundaries, and they will collate data locally for within their boundary. They'll be involved in things like the planning process, providing data for ecological consultants. They'll be involved in local nature recovery strategies. They'll work with organisations like local wildlife trusts to give them data on their sites for their site management plans, etc. Then you've got the national recording schemes and societies. These are quite often volunteer run and they will focus on a taxonomic group. So, for example, the UK Ladybird Survey, maybe no surprise to you all, that focuses on ladybirds. Some of them have quite a wide taxonomic coverage. So, for example, the Botanical Society of Britain and Ireland covers all vascular plants. Um, and then you've got other groups that are very specific, like the recording scheme that I run, the National Earthworm Recording Scheme. We focus on 33 species of earthworm that have been recorded in the wild in the UK, Ireland, and Channel Islands. And they will collate data at a national scale. There may be some sharing of data that goes on between local environmental record centres and national recording schemes. Data sets are obviously used for different things. With national recording schemes, it'll be much more about things like national distributions, conservation status assessments, maybe creating atlases, contributing towards information for identification guides, etc. And then you've got local natural history groups, which quite often will bridge the gap between these two kinds of organisations. So the example here is the Bishop Barnall group, but it might also be quite regional groups like the Yorkshire Naturalists Union or the London Natural History Society. It can be local mammal groups, local amphibian and reptile groups, butterfly conservation branches. There's lots of different examples. Quite sometimes they will have a county recorder for a specific taxonomic group, which will work with both the National Recording Scheme and the local environmental record centre. Again, usually volunteer run. And then you've got other projects and citizen science surveys that maybe sit outside of these groups. So I've got, as an example here, Project Splatter, which is an initiative looking to map roadkill in the UK. I think it's since got a new name, although I love that name. So all of these different organisations will be collating data and using it for different purposes and potentially sharing it with one another as well. So because of and that is quite often done under the banner of the National Biodiversity Network and may be shared through platforms like the National Biodiversity Network's online atlas. And the way we collect data has changed over time. Traditionally, it would have been specimen collections, notebooks, etc. And it would have been very hard to collate that data. But as technology has progressed, we've got apps and online digital recording tools and apps. So we're able to collect a lot more data than we would traditionally were. Gone are the days of the recording cards and things like that. And now quite often data is or usually stored electronically. And that has created a lot more data. And it can make the data quite difficult to manage. So it's really important that when we're creating our data that it meets modern data standards. So the fair data principles say that all data should be findable, accessible, interoperable, and reusable. So essentially, we should be able to locate data and access it. It should be usable between different organizations and uses and reusable for however many different functions we want to use it. So that brings us to validation of biological records. If we want our biological records to be usable and meet the fair data principles, then we need to make sure that they're in a certain type of format. So if we go back to that data flow process, we can see we've got the process of validation and the process of verification as checks. And these two terms are quite often used interchangeably. However, they really do have slightly different functions. So I'm going to define what those different functions are now, and I will be using those terms specifically for those two functions going forward. And it's important to note that the tool we'll be talking about later, Record Cleaner, that can help towards both of those processes. So data validation specifically checks that the data is in the correct standardized format. So in terms of biological recording, this would be checking that we've got correct current species names, that we've got valid dates, and that we've got good references that are valid as well. In terms of verification, that checks that the data is likely to be true. So was the stated species really at that location on that stated date? Now, I think it's important to note here that when we're talking about Record Cleaner later, Record Cleaner cannot verify records, but what it can do is give us messages that help us look back at and reflect on our own data to help the human verifiers that will undertake verification checks when that data is submitted to a record center or through iRecord, etc. So we're going to start with validation. Why do we need to validate data? Well, this is just some examples of how one date could be entered into a system. And as we can see, there are loads and loads of different formats there. So what we do in terms of validation of data is make sure that the date is in a set specific format. And there might be a number of different formats that are accepted, but in the example here, we would take data from any of those and we would say, no, it needs to be in this DD/MM/YYYY format. And I haven't even taken into consideration here American dates, which can completely change what a date means. So if, for example, this had been submitted in the American format of 07/04/19. In British systems, that would be misread as the 7th of April. So it's really important that we get things in the correct and standardized format so that we know exactly what it means. And likewise, with a grid reference, so we've got a grid reference here, and grid references are one of the most common errors in biological recording data because people quite often misunderstand how to change them if they're shortening them, or they can, and this can really translocate where the record is. So if you give an incorrect grid reference, you're saying it's in a different place to where it was. So I've broken down how a grid reference works here. The green letters, they represent 100 kilometre square on the British national grid. The red numbers relate to the 10 kilometre square. The blue numbers relate to the 1 kilometre square, and the pink numbers relate to the 1 kilometre square. So if I'd accidentally missed off the last digit when I was entering this, I would end up with a 5-figure grid reference, which actually isn't valid. It doesn't mean anything, and it wouldn't tell us where the record is. So if we run this through a validation check, because it's only 5 digits, the system knows that grid references need to have an even number of digits. It would fail a verification check. If we look at the next example where I've entered on my keyboard the O in the grid reference as a 0, again, if we run that through a validation process, the grid reference would come out as invalid and it would fail checks. So if we entered either of these data into Record Cleaner, for example, it would fail them and we'd be able to look at that and edit it appropriately. If I put it in correctly, and it was run through a validation, it would pass the check. What a validation check doesn't do is it doesn't tell you that this grid reference is correct for that record. It just tells us whether it's in the correct format. For species names, another thing that we can check, Record Cleaner and systems like iRecord, NBN Atlas will use a system we've got called the UK Species Inventory, and this has a big long list of all the different species that we've got in the UK, and it has a list of common names, it has a list of scientific names, it has details of previous names so that things can all be standardized. bear in mind that we'll come to common names in a sec, actually. Uh, so common names are something that can be quite problematic. So I personally believe it's always best if you can to try and use scientific names in your data set because they are more standardized. So for example, here the term daddy longlegs can refer to the whole group of crane flies, which is several families within Diptera. It could refer to the daddy longlegs spider, or it could refer to the whole order of Apleones, the harvestmen. It might be that different people from different regions use the word differently. So bear in mind what you think is an established common name might not be when we're looking at it nationally. And it almost certainly won't be when we're looking at it internationally. Uh, we can also validate vice counties. So the vice county system is a standard set of borders we've got for Watsonian vice counties. So these were created to kind of combat the ever-changing county borders to do with changes in political boundaries, etc. So this standardized set of vice counties has been used in biological recording for a long time, and we can use vice counties that are listed to see if the grid reference is in the correct region as well. And Record Cleaner will look at vice county numbers as well, but we'll come to that again shortly. So, right, we've talked about how these different aspects can be validated. So now let's look at how we can use Record Cleaner to validate that data. So what is Record Cleaner? So Record Cleaner started life fair few years ago as the NBN Record Cleaner, and it was a tool, it was a downloadable tool for checking your data. Now, it hasn't been supported for some time, and there's been some recent investment into the online record cleaner, which is a new tool based on the NBN record cleaner, where you upload data to the online record cleaner, which is housed on the Biological Record Centre's website. And the link to that will be going in the chat, but don't worry, we'll send that out later. It's free to use, it's available to everybody. And it's quite a simple tool. You'll see that the first page within the online record cleaner is just a data file upload. So you can upload CSV or Excel files. Um, with the first row has to be a list of column names. So you click on choose file, upload it, and that'll upload into Record Cleaner. And what it does is two things. It will do some validation checks, so checking that your data is in the correct standardized format, and it'll do some verification checks as a second stage, checking that the data is likely to be true. And I'd just like to stress what I said earlier. It is not designed to do definitive verification on your records. So if you run that through, it will not tell you that your record is correct or incorrect. It will not tell you that it's been accepted or not accepted. It is there as a tool for you to look at whether there are things that mean that your data might need looking at in a bit more detail. And we'll come to the specifics of that later in the webinar. So what can it validate? First of all, like we've mentioned, it can validate species names. So it will check that they are in the correct format and current accepted names according to the UK species inventory. It will check dates are in the in an acceptable format. It will check georeferences. So it will look at things like a grid reference or a lat long. And if that doesn't make sense, it's in the incorrect format, then it will let you know. And it will check vice counties. So with vice counties, it checks the number of the vice county to make sure that it's an acceptable vice county number. But it will also cross-reference that vice county number with the georeference. And if they don't match, so if that georeference doesn't occur within that vice county, then it will flag that up as not matching as well. So it can be really useful. There are some nuances with that we need to be aware of. So there are things that it will flag as potentially incorrect when they're not, and we'll cover some of those examples later. For verification, it has a set of rules which have been provided by the recording schemes that will help you check for example, whether your identification might be correct. And it does that using a system of codes to do related to ID difficulty. It will check the phonology. So the phonology is when the organism was recorded. So, for example, it will flag records of butterflies that were found outside of their flight periods. It will also check distribution. So, has this species been recorded in this area before, in this 10-kilometer square? If not, it might flag that up. And rarity. So, there are other things that might mean that your data needs some extra checking. So, for example, if it's a very rare species, or if the species, there's a bit of taxonomic uncertainty about it as well. So, who is Record Cleaner for? Well, it's for any individual recorder wishing to run checks on their own records. So, if you've got a spreadsheet of your records that you're thinking of putting into iRecord or sending off to a recording scheme or a local environment record center, you might want to run your data through Record Cleaner to validate the data. First of all, make sure it's all in the correct format, prevent the data being sent back to you or failing to upload into one of those systems, and for checking things that may be correct or that may need further look or may flag questions for verifiers. It can also be used by recording scheme organisers like myself, record centre staff, and anyone else who receives biological records from others and wants to do a quick check on them and to see if they contain anything unusual, unlikely, or in an incorrect format. It could also be used to consider, it's also being considered by the NBN Atlas to be embedded within their tool for checking incoming data that hasn't already been checked. And it also, I should point out here as well, there is also a version of Record Cleaner on iRecord. It's different to the online Record Cleaner that does those automated checks as well. So as an API, as a tool, it can potentially be also embedded by other recording systems. And one of the reasons why the development was recently funded by Natural England to improve it is there's an awareness that there's a potential for really big data sets coming from bioacoustics monitoring, coming from other automated monitoring systems and DNA derived data that could put quite a lot of strain on in-person verifiers. So this could be used as a system for screening that data. So what does it need as a bare minimum for its checks? So this is different from what a biological record needs as its bare minimum because Record Cleaner is not checking everything. So it doesn't have a list of, for example, of all people in the UK, so it can't check our names are correct and things like that. So the things that you absolutely will need in your spreadsheet that you upload to Record Cleaner to make sure that it can validate the data. As a bare minimum, it will need a record key. So this is a unique reference number for each record. And these are quite often generated by things like iRecord, etc. Now, if you're gathering your own data, though, in a spreadsheet, you probably haven't allocated specific record keys because that will be done when it goes into iRecord. So why have your own independent system? And that's absolutely fine. What you can do when you're uploading is there's an option to select auto row number, and that just allows Record Cleaner to assign a number from starting with 1 so that it has a number for each line. So then there is date. So it will require a date in order to check it. And that can be an individual date or a date range, but we'll come to that later. It will need a taxonomic identifier, which sounds really complicated, but really that is just something that tells the system what it is you're recording. So that could be a common name, it could be a species name, or it could be what we call a taxon version key. And a taxon version key is a unique number for that species. And you get that from the UK species inventory. I wouldn't expect most recorders to be putting that in. A scientific name is completely valid. And what this will do is tell you what your taxon version key is for that species went out of your download anyway. So you don't need to worry about having that if you don't have it already. But if you were running an iRecord download through it, you would have that information. It will also need a spatial reference. So that will be something like a grid reference or a latitude, longitude information. So a couple of notes about dates. I mentioned earlier on about formats. So Record Cleaner is set up to accept quite a few different formats. So there's a link going in the chat from Joss there to the page on dates and what formats are accepted. So for an individual date, these are some of the examples so that it will accept. And you'll see where it does the slashes and the dots here. It will accept where it's a single digit date or month or the double digit date or month. So it will accept any of these formats. If you've got a range, so let's say you put a camera trap, well, no, that's probably a bad example. Let's say you put out a pitfall trap to collect specimens in your garden and it was out for 3 days. You don't know what specific date the specimen was caught on. You can use a range. And again, on the page that Joss has shared in the chat, that will also be in the link below the video if you're not watching live, you'll see there's lots of different formats that it accepts, and it's just about checking those. If you've only got a range of months, maybe it's some really old records that you've mined out of a publication that you're working on. So we do this sometimes in London Natural History Society, where we go through old publications that we've published many, many years ago, back before people actually wrote down full dates for their records, and they would just say that they recorded something in a certain month or year. You can put ranges in for that. And similarly, if you haven't got a date for some records and you've got a range of years, so you might, for example, know that a specific naturalist from the past recorded it, but you have to use their lifetime as the range from year to year. You can do it with just year as well. But the full guidance for this is available online, and I'll repeat it in the content that we put online after the live webinar. Right, okay, so rather than do it live and risk my internet issues, I've done some screenshots to show you how you would submit a spreadsheet to Record Cleaner. So when you click on Record Cleaner, the first page you see looks like this. You would click here to upload your file. I recommend CSVs rather than Excels because some Excel formats occasionally fail. So if you can save it as a CSV file, that's always better. And then once it's uploaded, you click on next, and that will take you to the second page. So on the second page, you've got these four different drop downs. And when you click on one of these drop downs, it will have all of your column headers from your spreadsheet. So you just match the column header to the field. So like I said, for unique record key field, you just put that auto row number and then date, whatever you've got your date field listed as etc. I want to note here that unique record key field is compulsory. Date is compulsory. For vice county and life stage, so life stage would be whether it's an adult, juvenile, an egg, a fruitling, whatever. You don't have to add them in. But if you've got them, it's worth adding them in. When you've assigned whatever you need to assign there, you click on next, and you get to the taxon field type page, so page 3. So from this, once you select the type of taxonomic identifier that you want to use, it'll open up some more submenus. So in the example here, I've gone for the taxon name, and then I'm going to use the common name field I've got in my spreadsheet. And then I click on next, and we have the same kind of thing again for spatial reference type. And I've told it I want to use a grid reference, and I'm going to be using lots of different grid reference systems. So British, Irish, and Channel Island, because I've got records from all of them in my data set. And I've told it here which column. So it's the column titled grid reference that has that information in. Then you get onto a page where it says optional fields, and these are all of the other columns that haven't been used in your spreadsheet so far. And you can tick which ones of these you want to use. Now, what's really important to note here is that Record Cleaner is not validating those fields. All that this does is add these to the output that you're going to get from Record Cleaner. So you only really want to tick fields that you think might be useful when you're looking at the data afterwards. So, for example, um, I'd added the vice county number for my vice county field, but I've also got a list of the vice county names. So I might want to tick vice county here, which is the vice county names, because then I can, if anything comes up incorrect, I can see if the name matches the number. I might also want to tick my comments box because that might have extra information in it about how I did the ID. So if something flags up as potentially incorrect, I might have that information there. So you select whichever ones you want to add to your spreadsheet that's going to be output, select next, and then that will give you the settings summary. So we can see here all of the settings, and the value here is the heading of the column in my spreadsheet. And then it's got all the additional optional fields as well. So when you've done all of that, you click on validate here, and you'll get a summary of what's happened with the records in your spreadsheet. So I had 5,941 records. They were all checked. 4,589 of them passed. So that means there was nothing in those rows, those records, that that wasn't in the correct format and couldn't be checked properly. However, 33 of them had warnings and 1,319 of them failed. So they did not meet the data standard criteria for at least one thing. So when I look at this here, 1,276 of them, there was no code or name for the organism. So it might be that there's no common name for that species, so that the field was blank. There was also some unrecognized vice county values, and there's a note here that the spatial references for Irish vice counties can't be compared to the vice county numbers. So if I then want to go on and start the verification process, it will kick out all of these. So if you want to then look at the verification rules, you need to go back into your data and fix these problems. Now, because I can see that there's a lot here that just didn't have a name, I'm actually going to rerun my validation process. But instead of using the common name, I'm going to use the scientific name. And when I do that, I get quite a different result. So this time, 5,830 of my records passed. So it's got rid of all those ones that just had a blank in the common name. So now I've got 55 that failed the grid references and 56 that had an unrecognized vice county value. So yeah, if you have errors, might want to edit your data and re-upload to complete the checking process. So before we go into record cleaner rules, what I'm going to do is after this, I then played around with my data and I made some of it incorrect on purpose. And I ran another check and lots more things come up as incorrect. And I set off quite a few different of these rules. So I'm just going to share that spreadsheet now. And I'm going to show you some of the things that came up that were incorrect. So this is my spreadsheet here with my records that's come out. What I've done to make it a bit clearer, I have played around with the formatting a little bit. Here we've got the ID numbers, we've got the species, the common name, grid reference, vice county number, the vice county name, the date, and my comments. What Record cleaner has given me is a taxon version key, so that unique reference number. It's told me the result of the check, and then there's a message here about what things have failed. So I've organized my spreadsheet so that all the failures are at the top, just for the purpose of showing you guys. And then I've colored in the things that have failed, so that we can compare them to the messages. So here we can see that this grid reference has failed. Now this is an Irish grid reference, and when I'd run this check, I'd selected the British grid referencing system. So it's told me that it's an invalid grid reference for Great Britain. So if I was going to fix that, I would just run the check again, but then make sure I'm selecting British, Irish, and Channel Island grid references, and that would fix that. Here we've got another invalid grid reference for Great Britain. And if I look closely at this, it's a 5-digit grid reference, which doesn't exist. Now, luckily, in my comments, I've put what the grid reference was, and we can see that the 3 has been omitted off the end. So if I add the 3 in there for when I run it through next time, it will pass the check. Here, I've failed with the Vice County number. So it's saying here, the location not in Vice County 12. So this grid reference is not in Middlesex, is not in Vice County 12. But if I look at the Vice County name, I'd written this down as Middlesex, and I actually know that Middlesex is 21. So that was just a transcription error, so I can fix that as well. With these ones, there's been problems with the scientific name. So for this one, name not recognized, crabra, the horner, should actually be Vespra crabro. So if I fix that, that would work next time. Aparetidia limicoli should be Aparetidia limicola. Again, just a transcription error. But I can see that the grid reference is also not in Great Britain, but that's because it's an Irish grid reference. So we've got that problem again here. Here we've got a message saying TVK on name required. So it's um, a blank cell. So I can see that it was the square-tailed worm. So if I look that up, I'll find out that the um, scientific name for that is Icenella tetraedra, and I can add that in. So it would pass next time. Here we've got a failure on the date. I just need to convert this date into an accepted format. So the 23rd of September 2021, if I change that like so, that would pass next time. And then this is where we have a little bit of an issue with record cleaner. We've got unrecognized vice county values. Now, some records will be, we might not know exactly what vice county they're in because that square will cover multiple vice counties. So it's where the border runs through. And I've downloaded this data originally off iRecord, and it's come up with 24/21 for Buckinghamshire/Middlesex. So it will always flag those up. So if you want to push them through to the next phase, you'd have to change your data so that it matches just a single vice county. And bear in mind that for each grid reference, Record Cleaner does have a specific vice county assigned to it with a vice county that makes up the largest portion of that grid square. So that is something that is a little bit of an annoyance in Record Cleaner. Right. So, and by the way, obviously the color coding is something that I've added in to emphasize things. It wouldn't normally be like that. You'd have to look across the data. But we can see now why as well. It's a good job I added in the comment as an additional field to let me look. Everything else here has passed, so we don't need to do anything with that. Right. So if we go back to my PowerPoint. Right, okay, so that's validation, fairly simple. It's checking things a correct format. We're then going in, editing them and fixing them. So now I want to talk a little bit about the record cleaner rules. So the record cleaner rules are designed by recording schemes to flag records that may or may not be correct or may warrant further investigation. Um, these are the current recording schemes that have set rules. One thing I will flag is these rules were set up a fair time ago. So some of them might be quite out of date. Um, and like I said, Record Cleaner hasn't been supported for a little while. This is something that UKCH are working on addressing. So we'll be running we'll be running a webinar for the recording schemes at a later date to try and encourage rules to be updated. Updating the rules takes a lot of time. Recording schemes are normally volunteer run, so out of date rules are something that we have to deal with. And please don't. This is not a reflection on the recording schemes at all. So let's go through some of the rules. In terms of distribution rules, we've got one, the 10 kilometre square rule. And how that works is if we take a species like the pyramidal orchid, it'll look at a distribution map of that species. So this is what the recording scheme would have supplied, a data set saying, we know that the pyramidal orchid occurs at all of these 10 kilometre grid squares across the British Isles. And then I go out recording and looking for pyramidal orchids. And I find it at a few sites in London, in and around London. I find it when I go back home to Cumbria on a trip. I go on holiday to Scotland and I find it up in the Cairngorms. And I also find it on another holiday, no, let's say a work trip down in Devon. So these are my records of pyramidal orchids. If I run that data through Record Cleaner, it will tell me that all of these records pass the 10 kilometre square rule because they're all of these records that species has been found in those 10 kilometre squares before. However, it flags these ones as failing the verification rule because it's never been recorded there. And then what that means, it doesn't mean my data is incorrect. It means I need to look at it and either I may have confused it with something else, or maybe I've put in an incorrect grid reference. But I need to check that because if I have found it there and I can prove I've found it there, then these are really important records because it's not being found anywhere near there. These are all hypothetical data points. I did not find the pyramidal orchid in the Cairngorms. I've never even been to the Cairngorms, unfortunately. Right, so then we've got phenology. So phenology, like I said, is looking at when a species occurs. And we can look at the period within a year, or so there's two rules here. There's the period within a year. So what days of the year, or what months, weeks, etc., are you likely to find this species? And that applies at a life stage level. So what time of year would you find it as a caterpillar, or what time of year would you find it as a butterfly, an adult butterfly? There's also a period rule where it talks about when this species was first recorded or last recorded in the UK as well. So this is really important for records of things that have arrived due to climate change or they've been imported, etc. So if we look at phenology of the marbled white, this is the phenology of the adult butterfly. It is a summer species, and you're very unlikely to find it in even late spring and early autumn in flight. So if this is found outside of those periods, Record Cleaner would flag that as as a record to require further investigation. It might be that you've recorded the caterpillar as an adult by accident, or it might be that you've entered a date incorrectly, or that you found it at a weird time of year. So it could be any of those things. Um, so, like I said, Record Cleaner, one of the new developments is that it has the ability to define different phenologies for different life stages. So, for example, to prevent caterpillars being flagged as out of the flight period for adult butterflies. So as we're working with recording schemes on new rules, we'll be able to implement this in a lot more detail. It's also worth noting that it has a close to and far from comment. This is a new thing for the new record cleaner. Close to or far from categorization in the comments. So when you look at what record cleaner tells you about uh, rules that have been flagged, it will tell you whether it was close to the time that that has been recorded before, close to the phonology, or far from. And it does the same for the 10 kilometre square rule as well. So the definition of close to, I believe, from the record cleaner rules, is if it was within 3 days of the known phonology of that species. So this means that your record could just be telling us new information about that species. And yeah, likewise, it's the grids, 10 kilometre grid square next to an existing record. Far from means it's outside of those. So if we take an example, if we recorded the marbled white here, we'd get in the notification of the rule being flagged, it would say that it was close to. So it means that it's not beyond the realms of belief, that reality, that this could be a correct record. However, if we've got a marbled white flying in April, there's something wrong. It might be that it's escaped from a butterfly farm. It doesn't mean that it's not a incorrect record, but it means that there's something odd going on. Maybe there's a sudden change in behavior because it's been really warm that year. Who knows? But it would flag it up. And at location, here we've got one that's in an absent grid square, but right next to others and far from, again, we're going into Scotland. So in terms of identification difficulty, we've got kind of two rules. One's not really to do with ID difficulty necessarily, but we've got the identification difficulty rule and the dataset impact rule. And first of those is recording schemes may have set up a scoring system for how difficult species are to ID. So here I've took the leafhopper and allies recording scheme, and it's a 5-point scheme with an extra 6th point for where no difficulty rule has been assigned for that species. So 1 is the easiest to ID stuff, and 5 is the more difficult, the most difficult. And we can see here in this example, 1 means anybody in the field with a bit of experience could ID these organisms. 2, you can identify it in the field, but you do need a bit of care. You need to look at things maybe a bit closer, and some experience would be good. 3, you're going to need careful examination of external characters with a microscope and good lighting. But the idea is perfect. It's relatively straightforward if you do that. 4, same again, but the characters are not as easy to determine, and you might even need to dissect. And then going to 5, you are going to need to dissect and look at genitalia. You really need experience to do this. So you should always retain your specimen, because the verifier is probably going to need to check it themselves. And what the verification checks will do are flag these things. So you'll know as a recorder what your ability is with these. So it might be that for a specific group, you'll need to look at anything that's 4 or 5. But if you focus on leafhoppers, you might not need to even go through any of the 5s because you're pretty confident with what you've identified. Dataset impact rules. This isn't an existing rule, but I couldn't do a presentation without including earthworms. So this is Heliogelis oculatus, a very rare species of earthworm, only recorded from a handful of sites across the UK. We don't really know much about its distribution. So we can put a different set of rules that flag up and say that this is a rare species. So this is just a warning that this is a rare species. A verifier is probably going to want to check this when you submit it somewhere else. We could put an even stronger message on saying records for this species must always be verified. And for this species, we'd probably do that unless you're a trusted recorder that has experience of recording this specific species. I want to have a caveat here that rules are not absolute. So they're some of the rules are out of date, changes occur with wildlife, etc. And I'm going to just go through a few case studies. So the tree snipefly is a species of Diptera, a species of fly that is a specialist, particularly its larvae is a specialist in deadwood, and you'll find it in ancient woodland habitat. If we look at its phenology, back before 2000, it was pretty much only recorded from 1 week in July. It was always found at the same time of year as an adult. As we move on from 2000 to 2014, we started seeing changes in its phenology. So it was getting recorded as early as May and with some activity in September, but a bit more of a spread. And then if we look from since 2014, it's now active throughout April through to October, with some even earlier and later records. And that activity is not necessarily a clear peak. It is quite active throughout different parts of the year. I mean, it may be that it has changed its life cycle, and there are two populations a year. I don't know, but there are differences. So rules change. So if we were still using the rules from 24 from the year 2000, we'd be flagging lots of records. So this shows you that phenology does change, particularly with climate change. Climate change is changing the phenology of a lot of species, but also sometimes species adapt, or their phenology might change based on differences or in the phenology of their prey species or host species, etc. So if we look at distribution of the snipefly as well, so we've got the version of Record Cleaner in iRecord. We've got the Record Cleaner based on the rules that were set back in the 2000s. And then we've got the other species records since. So if we look at the year 2000 for the tree snipefly, the clear circles are where the species, we have records for it that we use to inform Record Cleaner, but it's never been recorded in iRecord. And the dark black circles are where it's present in both iRecord and Record Cleaner. So we can see here that there are in iRecord, it's still not being recorded some places that we know. As we progress from 2000 to 2014, it's now present where the red dots are, so here and here. It's now present in places in iRecord that are not in the Record Cleaner rules. And as we progress to 2015 onwards, that has spread. So this shows the importance in keeping the rules more up to date, which is why there's going to be a big push to update the rules in the near future. And the tree snipefly is not an exception. So this is the wasp spider. And again, we can see that this is something that is spreading. And there are a lot of records here that would still be flagged in Record Cleaner. So I just want to stress that with these records, you shouldn't be alarmed if Record Cleaner is saying that the species you found hasn't been found there. It might be worth then just having a look on things like the Spider Recording Scheme's website to see what the latest distributions are. And to give you an example from my own records, this is a fence post jumping spider. The video on the right is my video of it. I am almost certain, if not certain, that it was what I thought it was. Partly because I have friends that are really good ethnologists that told me, yeah, there's nothing else that could be. But when I put my data into iRecord, it got flagged by the inbuilt Record Cleaner rules there as being outside of the range. And that's because I was recording it up here where it's new, it's in iRecord, but it's not in Record Cleaner. So yeah, just a friendly warning. So some key points about interpreting Record Cleaner rules. They're not absolute. Things change, exceptions occur. So don't instantly think that it means that your record is wrong. The rules are there to help you, not to argue with you. So don't get frustrated if there are rules there that have been flagged up that you know are maybe out of date, etc. Flag records may require more evidence for verification. So that there is a tool. If you're if you're getting an ID difficulty flag when you're running through Record Cleaner, it might just mean that you want to submit some extra photos of certain features to help the verifier check it. Flag records may actually be very important records. So if you're getting records that are outside of the phonology or the distribution um, that we know of, it might be that it's a valid record and it's just the first time that we've ever had that record in that place or time. And the rules are only as good as the data that informs them. So bear with us while we get those rules updated according to current data sets. And again, just another example to stress the importance of not thinking that your records are wrong. This is another one of my records, Armadillidium depressum, a type of pill woodlouse. And it flagged up when I ran it through Record Cleaner just now as being far from the known distribution. This record has been verified by the, at the time, National Verifier for Woodlouse, Steve Gregory. So it is an accepted record. It was far from the known distribution, but it doesn't mean it was wrong. Um, so I want to explain that we've got another webinar coming up aimed at the recording scheme. So this isn't publicly available. It's for anybody that verifies records. So if you would like to come to that, please drop us an email, info@biologicalrecording.co.uk, and we'll give you the details for that, for getting signed up. And this is going to be all about the Record Cleaner rules with encouragements for recording schemes to maybe set new rules, update rules, etc. Right, and then I just want to show you quickly what the output would look like when you run the data through the verification part of iRecord. So we've go back to our validation. So this is the data set that I validated. So when you progress this, you need to click on continue to verification and click next. Now, if I do that, it's going to throw out all of these records. So just be aware, if you want to check all of your records, you need to go in and fix any of these validation issues. So we do that, we tick the box, we click on next, and then it will bring up this screen. So there's an option to use all rules. I recommend we always use all rules. And there's an option to omit ID difficulty messages. So if you want to just check phonology and distribution, you might want to untick this so that you don't get all of the flags for an ID difficulty, because even if it's flagged as a very easy record, it will come up then. But I recommend ticking it because I quite like to look at the ID difficulty. So I'll click on tick and then I'll click on verify, and then it'll come up with where rules have been flagged up. Now, bear in mind that a failure is simply a flag as being a rule has been set off, and that includes difficulty level 1. So that's why you might want to untick that box. 1,290 records passed means that they haven't activated any rules. 342 had warnings, so they'll most likely be the some of the ID code ones, and 31 have failed. So if I scroll to the bottom of that, there's then an option here to download the verify file. So we're going to download that verify file, and I'm just going to show you a couple of examples. So again, I've reformatted the spreadsheet so that it's got some things that I want to show you on top. And you can filter them according to whether they've passed, failed, or that it's a warning. You'll get this column here, which has the ID difficulty and whether it's passed or failed, and then the messages which explain those passes or failures. So here, I've highlighted this one yellow. So this is just a warning about the ID difficulty. So it's telling us from the hoverfly recording scheme It's ID difficulty 2, can be identified in the field with care and experience, needs a good view or the netting of a specimen to check, but the specimen can then be released, may be identifiable from a good photo or a series of photos. So it's just a bit of information for us. Um, Here we've got some failures though. So this one has failed. It's a spider and it's failed because its location is close to the known distribution, so it's in an adjacent square. Here we've got a southern migrant hawker that is far from the expected period. So if I was looking through my data, this one, I might not worry too much because it's close to the known distribution, so I might have just found it outside of the known range. This one, I would definitely have another look, and I might look at current known distributions to figure out whether the rule's out of date or whether, sorry, this is extended period of phenology. I might look at the phenology of that species or get in touch with a counter recorder. It might be that what I've recorded is a larvae instead of an adult and it's miscategorized as well. But yeah, so that information there about far from or close to is really useful. All right, I'm aware of time, so I'm going to end it there and then we'll go to questions.